welcome to session 2 for microbiology and immunology. Can I just check that you can hear me okay? Perfect, thank you. Okay, so last session we didn't quite finish the slides on the immune system, so I'm just going to recap where we were up to with that before we carry on. So we had a look at the immune system as a whole. We had to talk about the barriers. We looked at the structure of the skin, and we talked about the mucosa, which we've met in several times in other units. So we're talking about the gut lining secreting hydrochloric acid and proteases, and the mucous membranes in the lungs and the respiratory tract, which secrete mucus that helps to trap any pathogens that we inhale. So the barriers and their secretions are basically the first line of defense. So the skin has secretions too. Obviously, we have sweat, and that's got lysozyme in it, which is a special enzyme which helps to break down pathogens, particularly bacterial cell walls. So that's the first thing we talked about. We had a look at the structure of the skin. Obviously, we've looked at the goblet cells in the stomach and the immune cells in the respiratory tract earlier on. So first part of the immune system are the barriers to infection. Then the next thing is the non-specific response. So this is what kicks in if those barriers are breached. So if we get a cut and the skin barrier is breached, if we inhale something that manages to get through the mucus in our lungs, or if we eat something that the bacteria that we ingest manages to get through somehow, then these non-specific responses kick in. So we can divide it up into 4 things, really: the cellular defense, the complement system, the inflammation response, and the temperature response. So the cellular response, we've talked about the macrophages quite a bit over the course because they're active in various systems, because they're circulating. They're blood cells, they're white blood cells, special type of white blood cell, so they can circulate. We find them obviously in the blood, but these white blood cells can get into the tissue as well via the lymph. We'll have a look at that in a little more detail later today. So these cells are circulating. So we've met these macrophages in various other systems throughout the course. So macrophages, and they are primarily for phagocytosis, which is the engulfing of the pathogens and then breaking them down with enzymes. Then we had a look at neutrophils, which also can phagocytose pathogens, but they also release chemicals called reactive oxygen species. And there's just a few different chemicals. You don't need to know the names of them, but hydrogen peroxide is one. There's another one called superoxide dismutase. There's a few chemicals. Basically, they release these chemicals in this collectively. They're called the reactive oxygen species, and this chemical release is called an oxidative burst, and that kills the pathogens. Any that are in the vicinity near to the neutrophil will get killed. And then also in the cellular, non-specific cellular response, we've got these natural killer cells. And what they do is they kill cells that have been infected rather than actual pathogens. So this is really good for viral pathogens, because viruses get inside a cell to multiply, and they'll take out the infected cells by punching holes in their membranes. So then they can't survive because water will move in, and the stuff that's meant to be in the cell will move out, etc. So that's the non-specific cellular response. Then the next non-specific response we have is the complement system, and there's a little video on that. You can watch the video actually if you want to, because it's in the slideshow pack which you've got access to. You can click on the link to watch the video. But basically what that is, the complement system, is a load of different proteins, and they're just circulating again in the blood. And when pathogen gets in, they will attach themselves to the membrane. They'll label up the pathogen for destruction. They'll punch holes in it eventually by this cascade of proteins. They sort of build together and it's almost like looking at a little Lego building coming together. There's piece by piece by piece of protein after protein after protein in this protein cascade. They build a little structure that can then punch a hole in the membrane. But what they also do, these complement proteins, they send signals to the cellular response. Things like the macrophages, neutrophils, will detect these signals that are coming from the area of infection, from the pathogen that's been labeled up with the complement proteins. They're called complement because they complement the cellular response. They're in addition to the cellular response. And then the third non-specific thing that happens is we have this inflammation response. So what this can do is it can dilate the blood vessels, which means all of these circulating immune cells, proteins, whatever, can get to where they need to get quickly. The inflammation response also increases the permeability of the capillaries. So that means things can get in and out of the capillaries easier, things like white blood cells particularly. So that helps. And they can also cause something called microvascular coagulation. And that's sort of blocking up smaller parts of the vasculature, the capillaries and veins and things like that, to block the infection. It will stop it spreading so easily then. It traps the infection where it is. And the fourth part of the non-specific response that we mentioned was the temperature response. So our temperature is increased because the macrophages that are finding these invading microbes release a chemical which is picked up by the hypothalamus, which will then raise the temperature. And that raise in temperature increases phagocytosis. So it increases the engulfing of pathogens, this temperature rise. So 4 things going on after those barriers have been breached that try and hopefully contain the pathogen that somehow got through the barriers. Then, in addition to that, we have a specific response. So we've talked about antigen presentation before. So what happens when we get infected with a pathogen is it's broken up and presented. Remember, the major histopathology complex helps to present these bits of the pathogen, broken down bits of the pathogen, to a specific blood cell, a white blood cell. And what happens then, there's 2 types of white blood cells that kick in, B or T cells. And if they've got the right receptor for the antigen, for the bit of the bacteria or virus that's been presented, it will rapidly divide and produce a load of clones. And then this will help it with the response. Now, some of these B cells, they're just B and T are just types of white blood cells. Some of these B cells can become something called memory cells. So these are cells that remember the infection. They stay in the immune system, and these are the cells that then get activated if you come across this infection again. These are the cells that give you long-term immunity. So these are the ones that get stored away after you've been vaccinated or after you've been infected and recovered, essentially. So these are the ones that give us long-term immunity to infection. And that's a specific response. It's specific because the antigens are presented, and then we respond to that, or our cells respond to that antigen by dividing and producing loads of clones. And some of these produce antibodies as well, which help with the infection control. So that's where we got up to last session. So I will see if I can get this into slideshow mode. There we go. And move on to the next bit. So, as I was just talking about, we can, follow an infection or vaccination, we become immune, and there's different types of immunity. So you can have active immunity to a disease, and that's if you've made antibodies and memory cells that help you protect against it. So you've recovered from an infection or a vaccination, and these B cells have made a few changes and turned into memory cells. So two ways really you can get active immunity, which is infection or vaccination. And remember, vaccination is when you are deliberately exposed to either a dead pathogen, so one that is inert or been made inactive, or you are exposed to bits of a pathogen, so just the antigens somehow. So attenuated, they say, um, attenuated or dead pathogen. You can get passive immunity to a disease as well, and that's when you're given antibodies that have been made by another organism. So a really good example of that is in breastfeeding, because antibodies from the mother are passed to the baby. So this passive immunity, but that doesn't last if you stop breastfeeding, which obviously you do at some point because babies grow up. That immunity doesn't last. They don't get all the immunity that the mother has and they have it forever. They only have it when they're really small. And the reason for this is as they get older, they develop their own immune system and they develop their own immunity and they recover from their own infections and they get their own vaccinations. In the modern world, obviously, it would have just been infections. That's how it evolved. But we vaccinate now, obviously. So that only lasts as long as you are breastfeeding, and you can only give the antibodies that you have as well. So it's not. I mean, yes, it's good, but it's not fail safe. It doesn't mean they're completely protected from everything just because you're breastfeeding at all. It is possible to be given an injection of antibodies if you pick up an infection. This is the strategy that's used in rabies infections. So if you've been bitten by an animal that you know has rabies or is likely to have rabies, you can give antibodies in this case because it takes the body essentially too long to make its own antibodies to fight the infection. But rabies is really quite slow to take hold. You've got a bit of a window if you've been bitten by a rabid animal to get some antibodies on board before it kills you. Because if you don't treat it, it will kill you. It's a really, really horrible disease, actually. So passive immunity is temporary, it's short term, it's not your own antibodies, if you like, so it doesn't give you long-term memory. You're not producing these long-term immunity because you're not producing these memory cells. And the active immunity is much longer. And the reason for this is the antibodies that you take on with passive immunity can break down, and there's no B cells to actually make new ones. So with babies that are very small, neonates, so very recently born, antibodies can pass from the mother's blood, from the fetus, to the fetus. And this is referred to as natural immunity because it doesn't actually require any exposure to an antigen. Artificial immunity is the term that's used to describe immunity that's been basically arises from vaccination. So it's artificially achieved by putting, some form of the pathogen, whether it's dead or attenuated or what, in. Let me just. Sorry, I thought there was a message in the chat then. Okay, so a couple of questions then before we move on to the next topic. I'm not sure how easy this will be because we've just sort of jumped straight back in. But have a go, see what you can work out, and I'll pop the answers up for you in a minute. I think the last two are not too bad. Perhaps the first two are a little bit tricky. Okay, a couple of answers up. I will pop the answers up for you. Okay, we've got quite a bit of agreement on some of them. So that's a good place to start. So active immunity, it's vaccine derived is the example there because those are where the immune system has produced its own active immunity. The immune system has to produce its own immunity. And which of the following is an example of, I've just got to move a little box. Passive immunity is when you get an intravenous infusion of immunoglobulins. So if you've had an infection or you've had a vaccine, you're producing your own immunity. No, immunoglobulins would the first one wouldn't be B as well. Immunoglobulins are antibodies that have been given into the intravenous into the vein. So intravenous antibodies there. What type of immunity lasts longer? It's active because you can produce your own memory cells that can then respond the next time you get an infection. Passive just hangs around temporarily. And vaccines contain dead or weakened pathogens. I think you all got that one. So well done there. Okay. Right, that is the last slide on that chunk from last session. So I will just put the next ones up. One sec. Right, hopefully you can see that okay now. So this is the session 2 content. What we're looking at in this session is antibiotics and aseptic techniques and a little bit of the lymph as well. So the lymphatic system and what that does to support the body's immune system. So I've got the box to move again, one sec. Okay. So a couple of sessions ago, we were looking at the heart. We had a look at the tissue fluid and the lymphatic fluid. And we said that the tissue fluid or interstitial fluid is the proper name, is at lower pressure than the blood. So it can't return to the blood vessels very easy. Tissue fluid is therefore having to find its way back to the blood. And it does this by draining into the lymphatic vessels. And once it's drained into the lymphatic vessels, it's referred to as lymphatic fluid or lymph. So all of this stuff is essentially blood, but without the red blood cells. Once it's going around the tissues, it's called interstitial fluid or tissue fluid. Once it gets into the lymphatic vessels, that's what you can see in green on the diagram. Once it gets into these vessels, it's referred to as lymphatic fluid or lymph. And this fluid is moved through the lymphatic system in these vessels, eventually up to the subclavian vein, which is underneath the clavicle, which is the collarbone up the top by your heart. That's where the blood is at its absolute lowest pressure. Because if you think about the heart circulation, it pumps the left side is pumping under really high pressure. That blood goes all the way around systemic circulation, and then it returns up here, just under the collarbone, to the right side of the heart. By the time it's getting back to the right side of the heart, it's lost all of that pressure from that pump of the strong left side. So it's under really low pressure by the time it returns here. So that's the point, just under the collarbone, the clavicle, the subclavian vein can return the lymph. The lymph can be returned to the subclavian vein there. So that's how we get the fluid back. because it's such a low pressure area. Because, well, we talked about when we were talking about the circulatory system, we talked about the fact that the diameter, the combined diameter of the capillaries was huge. It would be sort of massive compared to the veins and the arteries, which it was in prior. The arteries first, so it goes from that to that. So moving into that massive cross-sectional area of the capillaries really slows it down. So it's under much lower pressure, and it can, by the time it gets back to the heart, it can receive this lymphatic fluid from the lymphatic system just beneath the collarbone there. So I'm just going to show this video, and I know you haven't got any sound, but I can tell you what's going on because it's a good one of the lymphatic system. Right, apologies for this. I'll just skip it. Okay. So there's the vessels in green. This is where the lymph is circulating. So those are the nodes. They're just clumps of lymph tissue, and that filters the lymph. And this is showing where it gets back to the bloodstream. So this is where the interstitial tissue fluid forms. It's leaving the capillaries because of the pressure. So a lot of this is just reabsorbed, or otherwise we'd have no blood if you think about it. A lot of it just goes back in. And it ends up in our veins. But the stuff that goes into the interstitial fluid, the tissue fluid, ends up here, back in the lymph. So those endothelial cells there are really loosely packed. There's really big gaps in between them, so pathogens can move in, which is a good thing because this is part of our immune system, but so can blood cells and things like that. So they're really loose junctions between the cells in these lymph vessels. So this is the lymph collecting vessels where the lymph nodes are found. So that's just the structure. You don't really need to know that. What you do need to know is it's full of white blood cells and it's an antigen presentation site. So the immune cells here will destroy the pathogens, or they will hold them there until they can mount an immune response. So that's the efferent vessel that's leaving the lymph node. There's valves in the lymph that are similar to the veins. So the lymph vessels contract to push the lymph along. And then there's valves to stop it back flowing. So, so there you can see the sort of larger structure of the main regions that are covered by the lymph nodes there. And then they'll all collect from one region into one collecting duct. So those are just the two main regions that drain. Okay. All right, so let me get back to my slides. Okay. So what's the main role then of the lymphatic system supporting the body's immune function? So one of the things it does is it transports this lymph fluid, as we've just seen. The lymph fluid contains the immune cells. It's collected from the tissues where it's called interstitial fluid, then called lymph, transports it through these vessels, and these contain white blood cells, a lot of lymphocytes, which are really important for detecting the pathogens while they're in the lymphatic system. So it's a really good sort of place to filter out the pathogens and screen out the fluids that's essentially going to go back and make up our blood. So think of it as like a really big filter to get anything out that has escaped all of our defenses thus far and shouldn't be there. So we've got these nodes, which you could see on the video and you can see on the diagram here, this sort of lumps, and these are the sites where antigen presentation takes place. This with all of those blood cells, white blood cells, the defense cells were packed in. They're filtering the lymph, they're trapping bacteria and viruses and any harmful particles that show up in those places. Also, the lymphatic system, we say that it houses the immune cells, so it's where you find a lot of them. They are circulating and they can move around, but you will always find a big supply of them here, and it produces them. So there's lots of lymphatic organs. The thymus matures the T cells. The bone marrow is part of the production of lymphocytes. The spleen, that's part of the lymphatic system, filters the blood. The tonsils and the adenoids are also really important to protect against anything that you inhale or eat as well. So they're all parts of the immune response. So when pathogens are detected in the lymphatic system, the lymphocytes are activated, it triggers the production of antibodies, it will coordinate with the immune response throughout the body, so it will just trigger so many other immune pathways. As you've probably noticed from last week, there are so many separate things going on and they all tend to trigger everything else to be in high alert as well. You know, when we looked at the neutrophils and the macrophages and the temperature and the complement system, it all sort of triggers everything to go into high alert if any of them get activated. So again, this system, if anything shows up, will trigger everything into action. Often the lymph nodes swell during infection. You know, people say, oh, my glands are up, and they think they've got swelling in their necks. Well, you can see here tonsils, and people say, oh, my glands are up because they're ill. That's exactly what's happening. It is just an inflammation of your lymphatic system while it's trying to get an infection under control. So why are lymph node biopsies important for diagnosis? Well, obviously, as we've said, they trap pathogens. So biopsies can reveal bacterial, viral, or fungal infections. I mean, that is a little bit extreme, to be honest. You'd have to be pretty sick with an infection to have a biopsy, but some infections do get you pretty sick, like tuberculosis, for example, is really nasty bacterial infection. And it will help to determine if you can take the biopsy and isolate the pathogen, you can determine what the infection is. But really, they're really good source of diagnosing cancers because all of these immune cells, which have evolved to trap pathogens, will also trap cancerous cells as well. So biopsies are really critical for identifying some cancers. Lymphomas are a good example, and cancers that spread to the lymph nodes, like breast cancer really commonly spreads to the lymph nodes, lung cancer as well. So it can help oncologists, the cancer specialists, to determine whether your cancer's spread or not if you have cancer. So that's another way, another reason why the lymphatic system is important. It can also help you to determine the stage of your cancer. You know, they sort of say stage 1, stage 2, up to stage 4. So not only how far has it spread to the lymphatic system, how far it's spread, what the treatment options, help to inform on patients' prognosis. Breast cancer, colon cancer are examples where this is quite useful. They can help the cancer team to determine what treatment is the best option, whether surgery, chemotherapy, radiation therapy, or immunotherapy is needed, depending on how far it's spread, for example. And they can help to determine the intensity and duration of the treatment and help just get feedback on how well the therapy or treatment is going as well, help to monitor the treatment. So quite a lot of uses there, particularly with cancer, for the lymphatic system there. Right, so the next thing you need to know is how to use aseptic techniques in a microbiology laboratory. Now, this isn't going to be particularly easy because we're not in a lab. So it's a bit of a tricky one, but you will be absolutely fine if you go on to do a practical degree and you haven't set foot in a lab yet. Believe me, I've taught, well, probably thousands of students over the years who've come from different countries where they might not have access to a lab or whatever, and they're absolutely fine after a few sessions. So we're just going to talk through what we do in a lab. So the first thing that you do when you're working in a lab is disinfect the work surfaces. There should be next to nothing in the area that you're working, because everything that is there is a source of contamination, and you do not want to contaminate anything that you're working with. If you think about this unit, it's called microbiology and immunology. So if you're working with microbiology, you're working with some organism. Might not be a pathogen, but it quite often is, depending on if you, well, it won't be if you're in college, but if you're in doing research, you might be working with a pathogen. So you do not want to cross-contaminate anything that you're working with anything that somebody who's previously been working with has left around. So first thing you do before you start work is you thoroughly disinfect the work surfaces. So when I used to go into the lab in the morning, the first thing we would do, we had something called a laminar flow hood that we'd work in, and that would look a bit like a fume cupboard, which you can probably remember from chemistry in school. It's a little bit different. The whole back wall sort of sucks the air out. So the first thing we do when we go into the lab in the morning is we'd spray the whole thing thoroughly with alcohol, like 70% ethanol, for example. We used to use a cheaper version, but same thing, same effect. So the whole thing would be completely sprayed. Then we'd leave it for 20 minutes with the extractor fan on. Essentially, you know, the extractor fans you have above your oven and your hob at home. It's pretty much like that, but it just covers the whole back of the surface of the sort of fume hood type thing that you're working in. So you'd switch on that massive extractor fan. You'd also spray all of that, completely cover that with 70% ethanol as well. The whole bench space would be covered in it, and you'd switch it on and leave it for 20 minutes to evaporate. And also that would have UV lights on it. So you'd switch the UV lights on and obviously go away, because UV is a carcinogen, you know, it can cause cancer. So you don't want to sit under the UV lights for very long. So you've got completely sterile, clean surface. You've sprayed it all with alcohol to sterilize it, and you're leaving the UV lights on that should kill off anything that's been left around before you use the workspace. So that's the first thing. You would have PPE on. So here would be tied back, and your lab coat would be on. Your lab coat, if you're working in a microbiology lab, I'll see if I've got a picture of my lab coat, me and my lab coat somewhere, but it's got like a polar neck that clips down really high. Um, so it's up to there. So you're not going to be dropping. It's not sort of like the doctor's coat that you see or used to. I guess you don't see it so much now. Medics tend to wear scrubs, but in the old days, you used to see them in a white coat. It's not one of those lab coats. It's got a high polar neck and pop buttons down. So you'd wear that. You would have your hair tied back, and you'd have gloves on. Your hands would have been washed before you gloved up. And when you put your gloves on, you spray your gloves with the alcohol that you sprayed all the equipment down, and then you sort of rub them together so that your lab gloves are completely sprayed as well. Now, the lab coat would be in a sort of porch area before you go into the lab. So it shouldn't be removed from that lab. We used to have different color lab coats depending on where we were working, so we didn't mix them up. So we had blue ones, burgundy ones, white ones, yellow ones. So it's not all the white coats that you see in the movies. It's very, very colorful, very multicolored. Usually in lots of different labs, so you don't cross contaminate. You have your own lab coat with your own name in each lab that you work in as well. So they don't come out, they stay in the sort of porch area. One lab that I worked in, I was working with some like medical grade stuff that was used with patients. So the procedure that we had to go through there was insane. We had to have so many layers of PPE on. We had to have covers on our shoes. There was, a sort of 20 to 30 minute just dressing and scrubbing down protocol before we could even start work in there. It depends on what you're doing. It's really intense. We always, when we're working in a lab with microbiology cultures, we always work very near to the Bunsen burner flame, because what this does is it creates a draft, a circulating current. So anything that gets airborne should just be taken up in the draft from the flame and not inhaled by you accidentally, to help keep the area sterile. Obviously, if you're working with anything that's even remotely dangerous, there's different levels. And you have different levels of PPE with like negative pressure and stuff. If you're working with anything really dangerous like Ebola, the safety precautions are insane. And it doesn't even have to be anything that you would consider really dangerous. If you're working with influenza or COVID, so you think, oh, it's only flu or COVID, it's not that bad, you would be in negative pressure suits with gloved up, like you see in the movies. Anything that's an actual known pathogen, the levels that you have to go to are extreme. Right, all the waste is exposed into different containers, and then it is autoclaved, which is so it goes into a special bin which is taped up, and then it's autoclaved, means it's exposed to really high temperatures and pressures, essentially like a huge pressure cooker. So really hot and really high pressures, and that will kill anything else off that's managed to get through all these sort of safety loops. And then any sharps have to go into a special sharps bin. So you'll have seen them at the doctors. If you've had a vaccination, they have special bins that they put the tips in. So any items that you use that are sharp go in those bins. So you use scalpels quite a lot in biology. What else do we use? Slides, microscope slides, they go in the sharps bins because they're so brittle. They really can cause injury. So that's before you start, really, all of that. Then we have to sterilize the equipment. So everything that you use is dipped in alcohol, and we use these loops, inoculating loops, quite a lot in microbiology. And what we use them for is to pick up a colony of bacteria, and then we scrape it either onto an agar plate, so a little sort of jelly plate, or we dip it into essentially a bit like a plastic test tube with a lid on. They're called universal tubes. They're the ones that you have to give a urine sample in the doctors, if you've ever given one of them. we use them in the lab to grow bacteria. So what we do if we're moving bacteria around is we inoculate with using one of these loops. So the first thing we do is we dip it in ethanol, which is highly flammable. Then we burn the ethanol off in the flame. So any bacteria which is contaminating this inoculating loop will get burnt off. And then we can use this loop to pick up our bacterial colony and transfer it to our, well, wherever we're transferring it, another agar plate or into some sugar to grow. So this is what we use to sterilize our equipment before we use it, as well as we use it to sterilize our waste afterwards. We use the autoclave. So we use a lot of reusable glassware like test tubes and beakers. Obviously, we use a lot of disposable stuff as well. The tips on our pipettes are always disposable, but the glassware that we use is reusable because obviously you can't just keep chucking everything out. It's not very good for the environment and really expensive. So lots of glassware, beakers, conical flasks, test tubes, boiling tubes, they all get autoclaved. So how we know they've been autoclaved, so it's high temperature and high pressure, is we wash them in the washing up room. You spend a lot of time washing up when you're a scientist. It's like half your job pretty much. You wash them, you put them on the draining rack so they dry, and then you put them into autoclave. So essentially they go in, I don't know, usually just put them in wrapped in foil, and then you put this tape on them. And this tape, once it's been through the autoclave, so high temperature and pressure, these little white lines that you can see here, they go black. So you know it's been through and you know that it's sterile. So you just unwrap it out of the foil then to use each time. So these are the pipettes that we would use in a microbiology, biochemistry, molecular biology, whatever lab. So there's a range of pipettes there, and these tips on the end are disposable. And each time you pick up a liquid, you hit the little, after you've used it, you hit this little button here on the back and it ejects the tip, and then you pick up a new tip. So there's three because there's more than three really, but these three are different sizes. So this one will go from 1 microliter to 20 microliters. This one, oh no, this one goes up to 20 microliters. This one goes up to 1 microliter, I think. And then this one goes up to 200 microliters. So really small volumes, like 200 microliters is a fifth of a milliliter, so a fifth of a centimeter cubed. But it allows you to be really accurate, particularly in molecular biology. But these are all autoclaved in a little plastic box, and you probably have a box of, I don't know, 200 tips of each size, and you know that they're sterile then. So when we're actually working with the bacterial cultures, so by that I mean bacteria that's been growing in a broth full of sugar and nutrients, what we do when we open it up is we flame the neck of the bottle, so literally pass it through the Bunsen burner flame immediately after open it and just before closing it. So any microorganisms that have got trapped around the opening, because they're like screw cap, should be killed off, hopefully. And it also creates an upwards current as well, stopping anything that's in the air from entering. Any Petri dishes that we're using, to scoop off cultures or test tubes that we're using, we don't leave them open. They should have lids, and we only open the lids as we're using it and then close it. We do not put the lids down. We keep them in our hands while we're manipulating whatever we're doing. So we don't put it down on the bench to transfer anything. So if I was going to do something with my Petri dish, I'd take the lid off, I'd use my inoculating loop, and then I'd put the lid down. I wouldn't sort of take it off, leave it exposed to the air, put it down. I'd just lift it a small amount, inoculate, and put it back down. So we're trying to avoid exposure to the air of all of our cultures because we don't want stuff from the air getting in them. This is a really big problem actually if you're dealing with mammalian cells, because everything that you breathe on it or shed in from your skin will contaminate it. So if you're dealing with trying to grow mammalian cultures because you want to do some experiments on obviously mammalian cells to see how it would, whatever you're doing affects humans. You really don't want any microbes getting in there. So, and then if you're dealing with microorganisms for whatever reason, you're researching them, you don't want to be inhaling them because the chances aren't going to do you much good. So we have to be very careful exposing the air to the plates that we're using or the media that we're using. You have to avoid any non-sterile contact. So your lab coats aren't sterile. You're not getting a new clean one out that's been autoclaved every time you go in the lab, but they're not removed from the lab. So they're not sterile. So quite a lot of people will spray down their lab coats before they go in. In the lab that I was working in that had medical grade stuff, my clothing was sterile every time I went in. It depends what you're doing really, but generally they're not sterile. So you don't want to be having things touching your lab coat, for example, because it will then potentially contaminate what you're working on, and what you're working on will contaminate your lab coat as well. So lots of keeping stuff separate going on. Right. Any questions on that stuff? Because obviously it's all very theoretical for you guys, I realise. I think you're all good. Okay, so next thing we need to look at is investigating the effects of antibiotics on bacterial growth. So antibiotics are chemicals that either slow down or stop the growth of bacteria. They don't work on viruses at all. It's kind of irrelevant when you're talking about viruses. They just don't do anything. So what I've got here is a diagram of an agar plate, which you may or may not have seen in school, depending on what sort of work you did in the school you went to. So essentially what this is a Petri dish, and the agar is it's just like jelly, like jelly and ice cream. Essentially it's a jelly-like consistency. It's got loads of sugar and nutrients in it, just stuff that helps the bacteria grow. It's basically just a sugary gel. And what we would do if we wanted to have a look at bacteria is we'd spread it out on the Petri dish and let it grow. Now, if you put loads and loads of bacteria, really concentrated amount onto the Petri dish, you wouldn't just see little dots of colonies, that you'd have so many colonies that it would they'd all merge into one. And that's what you can see on this agar plate here. There's hundreds of thousands of little colonies. And these spots here, these yellow spots, these yellow discs, they're about the size of, if you use a hole punch and the little discs that you get that come out if you use a hole punch, they're about that size, and they're made out of sort of like little bits of card. And they're soaked in antibiotics, pre-soaked, that you're not picking up wet ones unless you're doing your own experiment, but usually they're pre-soaked. And you can just pop them on the Petri dish. And if you get what we call a zone of inhibition around your disc, so here there's a zone of inhibition, the growth of the bacteria has been inhibited in this area, and here, and here, and here, and here. There's a big zone of inhibition here. So if you get a zone of inhibition of growth, that antibiotic is preventing bacterial growth in that zone. So you can say, whatever's on this disc is an effective antibiotic against this bacteria. So same for this disc, same for this disc, same for this disc, and same for this disc. All the other discs, whatever they've been soaked in, is ineffective against this bacterium. So this is one way to test loads of different antibiotics against a strain of bacteria. The bigger the zone of inhibition, the more effective that antibiotic is at killing off that strain of bacteria. So the most effective antibiotic that's been tested in this example is this one here. So here's a sort of look from above. It'll only take 24 hours really for the bacteria to grow. They grow very quickly. So what we would do is we would spread first the bacteria on Petri dish, not using the inoculating loop. You use a little bit of glass, which is sort of flat on the bottom. So you can just spread it out as if you're sort of rolling over it. So you'd spread it out over your plate, and then you put your antibiotic discs down. So the antibiotic discs, as I said, are pre-soaked with antibiotics, and they've got a little bit of writing on them as well to tell you which antibiotic it is. Then you'd come back after 24 hours. You put your Petri dish in an incubator, which would grow depending on what you were looking for. So if it was a bacterium that infects humans, so you'd probably incubate it at about 37 degrees because it would have evolved to live at human body temperature. So you come back after 24 hours and you have a look. And if there's an area where there's no bacterial growth, a zone of inhibition, that means that antibiotic is effective. So here, antibiotics B and C have worked, C working the best, A is ineffective against that bacterium. Now, it is worth checking back at 48 hours and 72 hours, because it might well be that A, which was ineffective, or so we thought, we come back later and we can notice a difference. So A is ineffective. That's not going to change if it's actually grown. But we might notice like there's a bigger, well, it might change. You might kill some off slowly, but probably not. But with C and B, you might notice the inhibition has increased. So this is what it looks like in real life. So this is bacterium called Staphylococcus aureus, which you've probably heard of before, because the Staphylococcus aureus is the SA bit of MRSA, which you've probably heard of, because that's a superbug. So those Petri dishes, which have been inoculated with Staph aureus, that's Staph aureus, that's E. coli, and that's E. coli. And they have these various discs here, which have inhibited growth. So they're not actually antibiotics that we treat as, we take as medicine, but they're things that inhibit bacterial growth. Now, one thing that gets in the news a lot is antibiotic resistance, because as we saw, sometimes those antibiotics aren't effective in preventing bacterial growth. And that's because antibiotic resistance evolves in bacteria. It evolves naturally because bacteria have tiny little called plasmids, and they can share them between each other. So random bacteria that they just bump into in the hospital lab, they can share these bits of DNA. And these bits of DNA contain antibiotic resistance genes. And that's a problem for us because we use antibiotics to stop and slow the growth of bacteria. But by using antibiotics, what we're doing is we're creating a selection pressure, which means that if bacteria have the ability to survive an environment with antibiotics in, they are more likely to reproduce. The ones with antibiotic resistance, obviously. And that's really good for them. So not good for us. We're creating a selective pressure that promotes antibiotic resistance by overusing antibiotics, by not completing the course. So some bacteria end up surviving at the end because we stop taking our medicine because we feel better. A few will survive after now got some little degree of antibiotic resistance. And then if enough people do that and enough bacteria share these antibiotic resistance genes, which is extremely easy for them to do, only the bacteria with the characteristics are able to survive and reproduce. These are the ones with the antibiotic resistant bacteria. So it's a massive problem in hospitals. You might have heard of C. difficile, which is Clostridium difficile, or C. diff. That's a very common hospital-acquired infection. Staph aureus, a very common hospital-acquired infection as well, because hospitals are obviously environments where there's loads of people on antibiotics. And these antibiotic resistant genes being shared around between bacteria, not helped, I should say, by medics and their person-to-person sort of spread, because they don't have a different lab coat for a different room. They just wander around in the same scrubs or lab coat every day. When in a hospital I used to work at, the scientists would be leaving their lab coats at the door and the medics would be in the cafeteria in theirs. And you used to think, no wonder there's a hospital-acquired infections going around. But yeah, so massively helped by these small circular pieces of DNA called plasmids that carry antibiotic resistant genes. They don't even need to reproduce to pass these on. They just need to come into contact with each other and they can pass them through specialist structures. So a real problem. And unrelated species, they don't even need to be the same species. What can happen with this is you end up with antibiotic resistant superbugs. And these are bugs that are resistant to several antibiotics. So if a bacteria comes resistant to one type of antibiotic, you can just treat it with another. But if they become resistant to all common antibiotics, there's one called methicillin, which is usually used as the last resort. And that's why MRSA, they call it methicillin resistant Staph aureus. It's resistant to methicillin, which is the last resort. That's why it's got that special name, MRSA. Once bacteria are resistant to all the antibiotics, we call them super bugs. And C. diff is an example, and obviously that MRSA that I was talking to you about. There's a little video you can have a look at here. This is what we are resorting to a set of time, but if you want to have a look, we have been resorting eat the dead bacterial infected tissue when we have antibiotic resistant patients, well, bacteria in patients. The patients aren't antibiotic resistant. It's the bacteria. So if we get to a scenario where people have bacterial infection, have been resorting to using maggots to eat away that tissue and the associated bacteria. So I won't show you the video because we're almost out. But if you have any, you can grab this video. It is in the course notes if you want to have a little look at it, but it is worth a watch. And actually, I should say, we were running out of antibiotics and it was quite worrying. It is probably going to help us and has been helping us a little bit to which are effective recently. So there is a little bit of hope because we were running out and it was getting quite scary what we were going to do. So any questions, guys, you can let me know. If not, I will see you on Saturday. What do you mean as in too frequently? I'm not, what were you talking about then? No, it's not good to take regular antibiotics because what you're doing, you're increasing the selection. Yeah, you're increasing the selection pressure then on the bacteria. So if any survive and then you hit them with another bout and go on then, let's see if you can do it this time. Let's see if you can do it this time. You just keep on keeping that same selection pressure up. But also antibiotics are often quite broad spectrum. So not only do they kill off whatever you're infected with, they're not good for your natural bacteria. Yeah, even with completed cycle, depends what your I'm not saying don't take antibiotics, obviously, because if you're sick and you've got an infection, it's better not to have the infection. But if you can avoid the infection, it's much better, is what I'm trying to say. If you can use, you know, if you can avoid getting sick, it's much better. And obviously, you have a lot of bacteria that should be bacteria all through your gut, which has a really important role in helping you digest your food, and immune system, which we don't even fully understand. And we have a lot of bacteria that lives in the female reproductive tract, which is important for maintaining the correct pH there. We don't want to be killing off this bacteria every time we have a course of antibiotics to kill infectious bacteria, because that's meant to be there and it helps keep our body in good place, homeostasis, where it should be. Everything. Obviously, if you're sick, it's better. You don't want to not take antibiotics, but some other way, it's probably best. It should be used as like a last resort. So a lot of people used to, when I was a kid, just have antibiotics. If you had a sore throat, you'd go to the doctors and they'd just give you antibiotics just in case. Or if you had an ear infection, they'd be, oh, have some antibiotics. But now they just say, it's fine, let it cover up, or clear up on its own. I know it's going to take better than over prescribing antibiotics. Okie doke. I will see you on Saturday then. Bye, guys.